Good morning, everyone. Let's wait for students to join and then we'll start off with our class. Okay, let's begin with our class. Other students who have joined. Okay, so we'll be discussing about the concept of Operon in this particular segment. First, we'll understand about that what is an Operon all about. That is, what are the basics that we had to make sure that they are clear before understanding the entire thing, and then we'll be understanding about the lag operon in this particular class, and we'll see how much we can cover up. And then there are two major operons, the lag operon and the trip operon. In addition to that, we also have ARA operon, which was this time asked like before 2022, like in June 2022. The question was asked out of nowhere. Before that and after that, no question has been asked. So when we'll be discussing about the PYQs in that particular segment I'll add few bits of this operon. Okay, so let's understand about what is an operon all about. Operon specifically, or just let's go with the help of explanation first. So what is an operon all about? Operon specifically, or in very simple terms, means that it is a arrangement of the genes in such a manner that it is helping in either the breakdown or it is helping in formation of any of the entity. Okay, that means operon is a regulatory entity which is helping in the formation or in the breakdown of the entire content that whatever we are looking into. So it is a unit of gene regulation. It is actually regulating a specific gene, right? And we can also call it as, there are like in the operon you'll see that there is a complete cluster of genes that are organized in a very sequential manner, and that is helping in the breakdown or the formation. Breakdown and formation have two very technical terms. First one is like the operon can be inducible or it can be repressible. Okay, inducible and repressible. Inducible, the word itself is suggesting its explanation, which we can induce from the external area or from with the help of any external component. We are inducing it, we are initiating the work. So inducible operons are catabolic in nature. Catabolic means that when you have any bigger component and it is getting cut into, or it is getting break down into small segments. So example for catabolic and the, or we can say inducible operon, is lac operon, which is your lactose operon. Okay, in this what is happening, the lactose is broken down into its smaller entities, into its subunits, with the help of the various genes that we have. Like the genes would be transcribed and then the proteins would be formed. So it is helping down in the formation of, or the breakdown of your lactose, and this is your inducible operon. It would be having, like lactose would be having positive and negative both regulation. That means the body does not require lactose. One more important point, lactose is, or sorry, operon is majority of the time applicable to your prokaryotes, and in very lower eukaryotes we have the operons, or the operon model is applicable to the prokaryotes and the lower level of the eukaryotes. Okay, so the lactose is, like, we do not need to break down the lactose for the complete life cycle. The complete operon should be, like, there should be positive regulation also, there should be negative regulation also. That means when it is needed, or whenever the lactose degradation is needed, it would be helping in what, it would be helping in, or it would be a positive regulation of your operon. Whenever the lactose is not required, we would be negatively regulating it. There's a complete story behind it, we'll understand it, wait for it. Second is your repressible. Repressible is your trip operon. Okay, so what is tripoperon? So tripoperon is helping in the synthesis of your tryptophan. Tryptophan is a amino acid. So it is again positively regulated and negatively regulated. In addition, it is a anabolic process. Anabolic process means, oh sorry, this is a anabolic operon. That means various components of the tryptophan, like whenever we need to synthesize the tryptophan, this operon would be working in the tripoperon would be working in. Again, the body does not require tryptophan operon for the, or does not require tryptophan amino acid for the complete life cycle. Like if any particular system is continuously working, there would be very much access of any, like over here itself, if the tryptophan operon keep on working, the cell would have a very high concentration of tryptophan, which could again be like cause any negative impact to the body, right? So having regulation for the repressible or the anabolic operon is again important, right? So positive and negative regulation in case of your tryptophan or the repressible operon as well. Okay, so this is the basics of the same. Then we have the main structure of an operon, like what are the various components of an operon. Let's look into this. We have a number of components of an operon. Let's say this is your one unit, this is your entire, like the DNA. On this DNA we're talking about one specific operon. Okay, so it would be having major, like one promoter would be there, operator would be there, and the structural genes would be there. So that means what, that means in addition we also have the regulatory gene as well. So this is your regulatory gene, which would be making the regulatory protein. Then we have something called as the promoter, which would be causing the initiation of the entire thing, right? Because over here now try to correlate, these are genes, they need to be forming their final product, and this would be formed with the help of same RNA polymerase working in, joining in, causing the transcription of the same and all those things, right? Over here as well same thing would be there. So you'll be having your promoter, where the lack RNA polymerase would be coming and binding. Then we have the operator, and last we have, these are your genes. Depending upon which operon do we have, the genes differ. Okay, so we have the structural genes. The operator is abbreviated as O, this is abbreviated as P, this is abbreviated as R, and these are your structural genes. Okay, so these are the main things that you have to keep in mind for the components. One more thing, when the regulatory protein would be formed, it would be helping in the binding, or like it would be helping in the positive or the negative regulation by binding to your operator. Like, we'll be understanding in the lag operon, you'll get a much better idea related to that, that how does a regulatory protein, like whether it would bind or in which conditions will it bind and in which conditions it would not bind. Okay, so this is about it. Quickly go through this. If you find any difficulty, just get the basics clear, and then we'll switch over to our main segment. Okay, just go through this, then we'll understand the theory segment of the basics itself. Just 1 minute, quickly go through this. Done, everyone? Theory segment of this, just let me know in the chat section if it's clear or not. Over here into C. And operon is what? An operon is a genetic regulatory system that is found in the prokaryotes, which is consisting of what? Which is consisting of cluster of the genes that transcribed together as a single mRNA molecule. These genes within an operon typically encodes for a protein with a related function. Like, we'll be understanding about the lab operon, it contains three main segments, or three main genes are there, that is Z, Y, and A. All these three would be getting like a specific protein would be released, a protein would be formed out of it, and on the protein, that specific protein would be helping out in what? It would be helping out in the, like, the complete, all the three proteins that get synthesized, they'll be helping in a very, like, coordinated result would be shown as a result of its functionality. So it is involved in a coordinated response to the environmental stimuli. So what are the basic components of an operon? Promoter, operator, structural genes, and the regulatory genes. What is the functionality of each of these? Look into it. The promoter is a DNA based DNA sequence that is located upstream of the operon, that serves as the binding site for RNA polymerase, initiating the transcription of the operon. Now, I am expecting you are understanding what an operon is all about and how does an RNA polymerase bind, what is upstream site, and all those things, right? So doing the specific topic in a specific area is very, very important, right? Now you can understand that if I would have taken this topic in unit number 2, you would not have understood this topic, that what is a lac operon, what is a binding site, what is operon, what is RNA polymerase. You would have not gotten the clear picture of the same, but here now everything is clear because we know the entire story behind transcription and everything, right? Now you have the operator. The operator is another DNA sequence that is found within the operon, usually located between the promoter and the genes. Look into the diagram. Operator is between the promoter and the genes, right? It acts as a regulatory switch that can control the expression of the operon by influencing the binding of specific regulatory proteins. Then you have the structural genes. So these are the genes that are the actual protein coding genes within the operon. They are transcribed as a single mRNA molecule, and they are typically involved in a related metabolic pathway or the functional process. Last is your regulatory genes. So in addition to the structural genes, some operons contain regulatory genes as well that are helping to encode for some transcription factors. They are helping in what? These are helping in the regulation of the operon. See, they can either enhance or they can inhibit the transcription of the operon in response to the specific signal. For example, let's consider you have lac operon. Okay, lac operon is doing what? It is helping in the breakdown of the lactose into its subunit. So let's say in the bacteria, lactose is not present. For example, lactose is not present. We are considering this. If lactose won't be present, then the operon need to be switched off, right? We need to inhibit the functionality of the lac operon. So in that particular condition, specific regulatory gene would be transcribed, and those proteins which get synthesized at the end, they'll be going and getting binded to the operator so that the functionality cannot be processed any further. Okay, this is very logical also to understand. Like, if you don't have okay, understand it this way. If you're not having Maggie at your home, no matter how many Maggie masala I give you, you won't be able to, like, make it, right? But, and on the other hand, uh, if you have Maggie at home, then there is a requirement for the masala also to be prepared. So in both the scenarios, depending upon whatever is required, we need to have either the inhibition or the enhancement. In the first scenario, when the main Maggie was not present, there was no need of initiating the complete process. There was no need of initiating the complete cooking process of Maggie, right? So that means you had to inhibit it. In the other scenario, when everything was in place, at that time you need to enhance it, you need to initiate the process. That means over here the enhancement need to be done. So are you able to understand this enhancement and the inhibitory thing? It depends upon your main requirement, whatever is required. If the lactose is there, it would have to be like the bacteria need to break it down to derive energy from it. But if the bacteria is not having lactose, there is no point having lactose operon in the active state. It has to be inhibited, right? Is it clear to all? Right, this is about the basics of the same. Now we have the two main things, the repressible operon and the lac operon, the repressible and the inducible. So there are two main ways. First one is inducible, second one is repressible. Understand this? Inducible operon, inducible means that they have to be initiated before getting the work started. Induce, we have to induce them. That means in normal scenario, in the day-to-day scenario, they are usually off, they are in the off condition by default, and they can be induced or activated by a specific small molecule which is given the name of an inducer, right? This inducer will be binding to the repressor protein. Okay, the inducer will be binding to the repressor protein, and when the inducer and the repressor joined with each other, that means what? We are switching on the operon, right? Initially it is in the off scenario, it is not doing the work. As and when the specific signal is taken up by this particular operon, and the stimulus is that you need to get turned on for getting the functionality done. So in that particular scenario, there occurs the synthesis of a small molecule called as a inducer, which binds to the repressor protein. The binding of the inducer to the repressor protein prevents it for binding it to the operon, to the operator, allowing the RNA polymerase to transcribe. That means simply just the inducer would be released, inducer would be synthesized, and it would go and bind to the regulatory protein. Then don't work, you need not to work in any manner, just sit and relax. Now the promoter is free, the operator is free, because if the regulatory gene, if any inhibitor comes and binds to the operator, what will happen? The promoter won't be able to do its functionality. Reason being, the RNA polymerase have to have a smooth road for moving ahead and transcribing the thing. But if, imagine the scenario, the operator, the regulatory protein, the regulatory entity comes and joined to the operator, that is a, that is working in the inhibitory manner, which is inhibiting it. So the promoter won't be able to do the work. Reason being, lack of RNA polymerase won't be able to move ahead in the scenario. So what will happen? It would be inhibiting it. So we need to inhibit the regulatory protein initially, we need to inhibit the repressor initially itself, right? So it binds, the binding of the inducer to the repressor protein prevents it from binding to the operator, allowing the RNA polymerase to transcribe the operon genes. Right, now example of inducible gene is your lac operon. In the absence of lac operon, the, or in the absence of lactose, the lac operon is repressed by a repressor protein. However, when it is present, it acts as an inducer, binding to the repressor and causing the operon to be transcribed. It's very, very like logical and basic to understand. When it is needed, it would be opened. When it is not needed, it would remain in the closed scenario. For remaining in the closed scenario, we need to bind it to the repressor. When it is not needed, in that particular scenario, the repressor need to come and bind to the operator. Simple, right? Are you understanding this main story? Because this lac operon and trip operon is completely based upon story itself. You need to have the entire flow of the mechanism clear in your mind. Is it clear to all that at which scenario it should be open and in which scenario it should be closed? We will be understanding about the complete lac operon also, but in the example format, is it clear to you or not? Just check. right? Second is your operons, means the operons which are usually in the on conditions, but we need to deactivate them, we need to repress them when a specific small molecule called as a co-repressor binds to the repressor protein. So the repressible operons, they are usually on by default. They are, and they can be repressed or deactivated when the small molecule called as a co-repressor binds to the repressor protein. Binding of the co-repressor to the repressor protein enable it to bind it to the operator and blocking the RNA polymerase and preventing the transcription of the complete operon. Example for the for the repressible operon is your operon in the bacteria, which is involved in the synthesis of your amino acid tryptophan synthesis. So when the tryptophan levels are high in the body, in this particular scenario, the operon need to be closed. The operon need to be deactivated, right? So in the case of deactivation, your operon, like the tryptophan, is itself working as a co-repressor, and it would go and bind to the repressor protein, leading to the repression of the operon. As in when the tryptophan is not present, the tryptophan won't be able to go and bind to the repressor, and then the tryptophan or the operon would be in the on scenario. So over here there's an additional thing which you have to keep in mind, that the tryptophan itself, whatever the product is it is itself working as a co-repressor. Okay, the regulation of the operon allow the bacteria for controlling the expression of the genes that are involved in specific metabolic pathways or the cellular processes in response to changes in the environment, and it is providing a means for efficient gene regulation and conservation of energy and the resources, right? So these are the two operons. First one is the inducible, second one is the repressible. So giving you 2 minutes for reading out the things, check it, and then we'll start off with our very first operon. This was about the basics of what operon is all about, and I'm expecting this much is clear to all. Take your time, read all this, and then we'll start with the main lab operon understanding. That particular thing should be right. Giving you the next slide for reading. read this very very properly, okay, because there are a lot of segments which you have to keep in mind. So just go through this, and if you find any difficulty you can ask. Done everyone? Should you lack Operon? If you have any doubt till here, you can ask. Others also, is it clear? No doubt. Okay, let's proceed further now. Lack Operon. Okay, we have already understood this terminologies. Now understand this. Lack Operon. That is lactose Operon. Okay. When you look into E. coli, there are two of the food sources. There are two of the energy sources that the E. coli can have. It could be glucose and it could be lactose. Okay. Now understand it logically itself. So there are two sources. glucose and lactose, right? Remember that glucose is a preferred source and lactose is a secondary source. That is, if glucose is not present, in that scenario lactose would be used, otherwise it will not be used. Okay, simple. Now, I'll give you few scenarios and you have to think about it yourself. In which of the scenario will the lactose operon be open and in which scenario lactose operon will be closed. Okay. Do not be in a hurry of marking it. Just understand it. Okay. This is for glucose. This is for lactose. This is for on situation and this is for off situation. Okay. Remember, glucose is a preferred source, lactose is a secondary source. Okay. In the first scenario, write down everyone. If glucose is present and lactose is absent, so what will be the condition of your lac operon? Think about it, everyone. If glucose is with the condition, everyone write it down. Eight of you are there in the class. Yes, it would be in the off condition. On it, the lactose operon will not be on. Second scenario, glucose is absent, lactose is present. What will be the scenario over here? Lactose present, glucose absent. Yes, in this scenario, it would. Now consider both of them are present. Now, what will be the scenario? Glucose and lactose, both of them are present. What will be the scenario? Yes, having lac operon being off because the glucose is there. So the E. coli would be having its preferred source first, right? Fourth scenario, what if both of them are absent? Now, what will be the scenario if both of them are absent? Think about it. Yes, it. So you can see which is the scenario in which it is being in a turned on state. Only one scenario when lactose is there, glucose is absent. In this scenario, only we have our lactose being the operon being in the on state. How does all of these things happen? We'll understand just now. But this was the basics of the same. So glucose is present. Glucose is the preferred source. Okay. And lactose is that if the glucose is absent in that scenario, only the lactose would be metabolized by the E. coli into glucose and galactose. Okay, the lactose is made up of, or it gets formed with the help of glucose plus galactose, right? So lactose would be broken down into glucose plus galactose, and then the further energy would be derived from it. The E. coli break down the lactose when available, requiring three genes. Which are the three genes? Now, look into the diagram, everyone. This is your inducer, this is your promoter, this is your operator, right? And this is your lac operon's main structural genes. First is your Z, then is your Y, and then is your A. Okay, Z, Y, and A. You have to remember this uh, sequence as well. Z, Y, and A. Let's understand about this and all the three genes. Gene Z, gene Y, and gene A. In addition to that, you have your promoter, operator, inducer. That is your regulatory protein or your inducer. Now understand this. Okay. So there each of these genes are performing a specified function. Okay. You have gene Z. It breakdowns the glycosidic bond of the lactose. So gene Z is doing what? It is breaking the glycosidic bond, okay, of your lactose. And over here, which linkage is getting broken? The linkage which is getting broken is beta 1,4 linkage, okay. Beta 1,4 linkage gets broken. So your lactose gets converted into glucose plus galactose. Clear? Now, yeah, this is the first thing. So we can say that gene Z is producing what? It is producing beta galactosidase because it is breaking this bond. So it produces what? It produces beta galacto G A. It is producing beta galactosidase enzyme, which helps in the breaking of the glycosidic bond of your lactose, helping in the conversion of lactose into glucose plus galactose. Okay, then you have gene Y. So, gene Y is producing permease. Okay, it is producing what? It is producing permease, which is acting as a carrier protein. Gene Y producing permease, which is acting as a carrier protein, which is responsible for the transposition of the lactose. That means for the transportation of the lactose. So it is a carrier protein helping in transport of the lactose. Okay, it is a secondary active process. Now I'm expecting you must be understanding the meaning of this secondary thing and the active process thing, and which is in relation to your transportation. Okay, so this is how you have to correlate the thing. We have discussed about this, the how does the transportation occurs, okay, and what is secondary active transport, primary active transport, all these things in detail. Third is your gene A, which is producing your trans acetylase. Okay, what is it doing? It is producing trans acetylase. It has no specified role in the metabolism of lactose. It is responsible for the detoxification means that when the beta galactosidase is produced, we might have some kind of uh, toxins also getting accumulated over there. So for that it is required transacetylases responsible for the detoxification. Okay, so it helps in the acetylation of the thiogalactosidase and causing the detoxification of the same. Okay, remember about this particular thing. So transacetylase, it is working as a donor of your acetyl group, donor of your acetyl group, and in the acetyl coenzyme A acetylation. So remember one thing, each and every pointer that we are discussing is very important. Okay, try to jot down the things. See, I have not added every small bit. You'll have to make sure that you are working hard on your end and you are noting down the important things also. Okay, so you have gene Z, gene Y, and gene A. Each of these has a specified role to perform, and collaboratively, three of them in a very coordinated fashion is getting the metabolism of the lactose done. Okay, just check over here if you have any doubt. Right now, let's understand your structure. Okay, so Z, Y, A, remember the functionality of each of them. So look over here, whatever I have explained there, same thing is written. Lactose operon has three of the utilization genes, LAC Z, LAC Y, and LAC A. LAC Z beat codes for beta galactosidase, which breaks down the lactose. In codes for beta galactosidase converts the lactose to the allolactose. Okay, remember the main thing of over here is that beta galactosidase would be converting the can also convert, like in addition to converting it from lactose to glucose plus galactose, it can also convert lactose into your allolactose. Okay, what is allolactose? So it would be working as the actual inducer means to initiate the main process. Then zy lac y, it is encodes for the lactose permeases, which allows the lactose to enter the cell. And then lac A, it enters encodes for the trans acetylase, which helps in the activation of thiogalactosidase by activating it. Okay, in adjacent to the structural genes, a common promoter is present. Lacopenon is turned on when the lactose is present and the glucose is absent. Remember this particular condition: lactose should be present and glucose should be absent for the initiation of this work. Okay, then yeah, when the glucose levels are high inside the lactose utilization genes, are not needed. Operon is shut down with the help of lac repressor, which binds to the operator, which is present adjacent to your promoter. Look over here in the diagram. The depressor would be binding to the operator, which is present adjacent to your promoter. So what is it actually doing? It is actually blocking the entire pathway of your RNA polymerase to move ahead. Okay, so lac repressor, what does it do? It blocks the binding of the RNA polymerase. What is the main functionality is it doing? It is actually blocking the binding of the RNA polymerases. So when the glucose level falls, lactose is detected in the medium, lacoperon is turned on and it produces the enzymes to break down your lactose, right? Now understand the entire thing. There are two main things what is actually happening. First scenario, in the case of your lactose absent. Okay, in case of your lactose being absent, what will happen? The repressor would be produced, right? Repressor would be binding to your operator, right? As in when it binds to the operator, RNA polymerase movement is blocked. RNA polymerase movement blocked. And as in when RNA polymerase movement is blocked, there is no transcription. That means we can say the lac operon is in the turned off situation. The operon is in off situation, right? Understand this entire thing. Lactose absent, repressor would be induced or produced. It binds to the operator, RNA polymerase activity blocked, and you have your no transcription done, because, or we can say the lactose operon being in the off situation. Second scenario, when the lactose is present, in this scenario what will happen? In the case of lactose working, this is your repressor, promoter, operator, Z, Y, and A. Okay. So now what will happen? The repressor usually, as a usual case, it would be producing your mRNA. It produces a repressor over here, right? Now what happens? This repressor is present. Over here we have the lactose outside the cell. This is your cell. This lactose enters into the cell with the help of what? With the help of permease, right? The lactose gets converted into allolactose, right? This allolactose will be binding to your repressor and causing the inactivation of your repressor. Okay, so what is happening? Lactose convert getting inside the cell with the help of permease enzyme, causing the conversion of lactose into allolactose with the help of beta galactosidase enzyme. Allolactose getting converted into it would be getting attached to your repressor and causing the inactivation of your repressor. As and when your repressor is inactive, it will not be going and binding to the operator, and the lacoperon will be in the turned on scenario. Remember one thing, whether the lacoperon is, even if in the is if even if it is in the off situation, even if it is not producing anything, it would still have a basal level. Okay, basal level of response would be shown by it, because in the worst case scenario it requires permease all these things, right? So how will it actually get it? So remember that lactose, be it lactose, yeah, in the lactose scenario there is basal level of condition or basal level of work getting done. So when you look into the transcription of your lacoperon, there are three ways in which the transcription of your lac operon is getting done. First one is the negative, that is which is in the negatively repressor. That means when you have your operon being in the turn situation. Okay, negative repressor, that means what? The repressor is the repressor is getting inhibited. Second one is your basal. Third one is your positive repressor, that means when the repressor is in the off situation. So that means what? Negative repressor means your lac operon is turned on. That means over here the work or the transcription would be more than the basal amount. Then second is your main thing, that your basal amount is there, no matter it is positive or negative, to keep the emergency work ready. And positive repressor, when your operator is in the, or when your operon is in the turned off scenario, over here there would be condition where less than basal would be produced. Less than basal means less than optimum concentration or optimum level of work is getting showcased by your operon. Okay, remember about these three things as well. With these three things you can understand this and this. Okay, just check if it is clear or not, because all of these things are very very important. So giving you just 2 3 minutes, go through this, then we'll proceed further. Done, everyone? Right, there is to this. Let's understand that. Right, should we continue? Now understand one more segment of the same, and which is related to, like, the glucose and the galactose. All of these things alone cannot do this work. Okay, they cannot do the work alone of binding to the promoter and getting the work done. There are two other entities which is given the name of CAP, catabolite activator protein, and second one is your CAMP, which is your hunger signal. Okay. Now, CAP and your CAMP, these two other entities are there which is working in the entire scenario. How? Remember one more thing, that glucose inverse proportional to CAMP. That means when glucose is more, CAMP would be less. When glucose is less, CAMP would be more. Okay. Now, for efficient working of your lactose operon, CAP also need to help in the process. Okay, consider the scenario when the glucose level is low. This means what? Glucose is low, that means there would be high CAMP, right? When glucose level is low, there would be high CAMP. Now, CAMP and CAP, they need to work in association with each other to get the work done. So CAMP is high, which now this CAMP would attach to your basal is quantity. Vagisha, basal is quantity, basal amount, like least or very minimal amount. That is what basal is, right? Base amount or the minimum amount possible. Clear? Vagisha, clear? Amount of lactose over here, some amount of lactose, there need to be some concentration of lactose so that we have some concentration of beta galactosidase, permease, and all the other things, so that this lactose can enter. Now just imagine this scenario itself. When the lactose is present, this need to be turned on. For needing to be for getting to it turn on, we need to have allolactose. From where will you get allolactose? Via conversion from the lactose, right? Lactose gets converted into allolactose via beta galactosidase. From where did we get this? We got it via the transcription of your gene Z. So from where did we get the gene Z? Because our lactose was working, or our lac operon was working in basal concentration. It was working in a very basal amount, so that some concentration of beta galactosidase permease is present in the cell for all the scenarios possible. Clear? Vagisha, is your doubt clear? Yes, when lactose is present, lac operon is on. Did you not join the class from the very beginning? We are building this concept from last 1.5 hours. When lactose is present, lac operon is turned on. There are three ways in which the transcription of the lactose operon is there: basal, more than basal, less than basal. There need to be a basal amount need to be like getting done. That's why you didn't join from the very beginning, na? That's why you're getting confused. So watch the entire recording first. So if you joined late, then for sure you'll be having the doubts, na? Then first join, like get the complete class done, then you'll understand the complete scenario, because we have discussed from, it's almost 1.5 hour, like it's 1 hour 12 minutes since we are discussing all this. So first get through all of this, and then you'll yourself understand this thing. Okay, let's complete this last chunk. Everyone understand this. When the glucose levels are low, so we were discussing that glucose and CAMP are inversely proportional to each other. CAP and CAMP, both of these are required for your better performance of your operon, right? Higher concentration, like when the glucose is low, high concentration of CAMP would be there. CAMP would be attaching to CAP, and these two would be attaching to your promoter site and helping in a higher transcription or higher level of the efficiency being shown for your entire operon, because glucose is less, lactose is more. In this scenario, CAMP would be more, it would be attaching to CAP. They both in association with each other, they both will be binding to the main site, and as in when they bind to the main site, it, the CAP will be helping the RNA polymerase to bind to the main promoter region, and then resulting in a higher level of transcription. Simple. Second, when the high concentration of glucose is there, there would be less of CAMP, less of CAMP would be there. It won't be able to activate, or it won't be able to join to CAP. They won't go and bind to the site of your their attachment onto the operon, and hence the lac operon would be staying in the turned off scenario, because your glucose levels are high in it, right? Simple. Very very simple. You just have to understand all of these things. Just remember this, that glucose and CAMP are in inverse relation to each other, first, and second, CAP and CAMP both join together, like both of these can work in association with each other only to get the higher level of transcription done. Okay, just check if it is clear or not. I'm sending one image also on the group, you can check. Would be easier, much easier for you to get the complete understanding. very low amount, just to make sure that the enzymes are present in a very basal concentration, that if as and when the lactose comes in high concentration, the work can be initiated. Okay. Clear everyone till here? Done this. Check over here. Absence of inducer and the presence of the inducer. When the inducer is absent, the repressor would bind to the operator, preventing the transcription of your lab operon. And when the inducer is present, so these would be transcribed, and the next set of proteins would be formed, right? So you have the absence of inducer and the presence of inducer. Then you have the catabolite suppression, what we discussed about the CAP and CAMP, similar thing. Then look over here, same thing, whatever we have discussed, exact same thing is there, it's just the depiction. I've already shared this same image on the group also. In the absence of lactose, lac repressor binds to the operator, transcription is blocked. However, in the presence of CAMP and CAP complex, RNA synthesis is blocked when the repressor is bound to the operator. Same thing, CAMP and this, but we have the operator being there. We have the repressor getting bound to it, even then the work won't be there. The third scenario, in the presence of lactose, the lac repressor is released from the operator and transcription proceeds at a slow rate. But as in when CAMP and CAP comes in joint, so it would increase the RNA synthesis. Okay, so it stimulates the RNA polymerase activity and it would be initiating the functionality of your RNA polymerase and increasing the speed of the transcription of the genes required. We've already done this. Before this, just go through this. If you find any difficulty, you can ask. The next table is similar to what we have already done. Right, this is something we have already used. Present, lactose absent, in what scenario what will happen. So see, when the lactose is absent, glucose is present, repressor will bound, yes. CAP will bound, no. Lactose, the transcription of the lactoperon would be done, no. Second scenario, lactose absent, glucose absent, repressor bound, yes. Cap bound, yes. Transcription won't be there. Third, lactose present, glucose present, very basal amount would be there. Fourth, lactose present, glucose absent. Repressor bound, no. Cap bound, yes. Transcription would be at a very high concentration. Okay, already covered this, nothing new to it. Then we have the mutations in the lac operon. Okay, this is something we are going to discuss in our next class because it's a complete set of new thing which you have to understand. till here we have covered about the basics of what LAC Operon is, basics of what Operon is, and the complete functionality of your LAC Operon. Okay. In the next class, first I'll either take the complete trip Operon, and then we'll understand the mutation of both of them, or I'll check whatever is more easier from a student's perspective. I'll do accordingly. Okay. So is everything clear to everyone till here? It's a very important concept, and this year also we got a question in December, in this June exam also. So we got the question related to the same. So I'm expecting this concept is clear, and majority of the time the question is asked uh, in which you have the operator of a lac operon or a promoter of a trip operon. They are mixing the two and then they are giving the question. So in that, what you have to have make sure that you are having clear understanding of both the scenarios, that how does a lac operon work, how does a trip operon work, and then only you can solve out the question related to the same, right? So this was the major concept that I have to take up in the morning class. Hope everything is clear. In the evening class, we'll cover up the tripoperon and the mutation cases as well, depending on the timing. And on Sunday, you all have a test. Make sure that you prepare well for Sunday's test. Okay, that's it. On Saturday, we'll be having general aptitude. Make sure that you come in each and every live class. And if you're not able to come to the live class, it's okay. Make sure that you're going ahead with the. We'll be doing it, Rekha. Don't worry. But I know each and every segment of it. Don't worry. You will get every single thing. Just have some patience. Okay. At this point, this concept is important. I have given that question for you all to study first. Okay. We'll be doing it when we'll do the previous year question session. You all know that after the, like, on the last day of the class, whenever we have general this unit getting completed, on that day you have a number of questions discussed. Okay, so I'll be taking up that question in that particular segment, so don't worry, right? So let's meet in the evening now, same timing, 6 o'clock. Make sure we are there on time. And from 16th of July, there might be slight changes in the timing of the classes. Okay, because I have two batches starting from 16th July, so there are chances I might change just 1 hour before or 1 hour after. Okay, I'll let you know the exact timing, but till then there is no change, same time would be getting done. Okay, hope everything is clear. It's meet in the evening now. Bye everyone, take care, study well, and any doubt, any difficulty you can ask. Okay, bye everyone.